Hello and welcome to Nikolai's genetics lessons, and this video going to be about forensic genetics, and today I have prepared two multiple choice questions for you, one multiple choice question and one true-false question. As usual, I recommend you to pause video here, try to solve each problem on your own first, and when you would be ready you can run video again and you can compare your answers with my answers and explanations. So here is the first question: which tissue samples are used for DNA fingerprinting? And here is the four answers to choose from. Answer A, hair; B, skin; C, blood; D, any of the above. And the correct answer is answer D. So any of above tissues can be used in order to extract DNA. But for example, hairs would be used mostly in order to extract mitochondrial DNA, because it is very hard to extract nucleus DNA from the hairs, because hairs represented by mostly dead cells with highly degraded nucleus DNA. But recently I read an article in which new technique explained, and nowadays scientists can also extract nucleus DNA even from cat hairs, because those hairs that contain root have skin cells, and it is not hard to extract DNA from the root of the hair. But nowadays, as you see, even from the cat hairs, scientists can extract nucleus DNA. And of course, skin can be used in order to extract nucleus DNA. And as for the blood, it is interesting, as you know, that about over 50% of the blood is serum. Serum is water plus some proteins, hormones, sugars, and another 45% are red blood cells, and red blood cells doesn't have nucleus DNA, so don't have nucleus and don't have DNA, and cannot be used in order to extract DNA. About 2% of the blood represent platelets. We need these cells whenever we cut a skin or have internal bleeding. The cells would help us or our bodies to stop bleeding, and those cells also doesn't have nucleus, and only white blood cells that represent less than 1% contain nucleus, and we use those cells in order to extract DNA. Fortunately, even one small drop of blood contains millions of the cells, and even 1% would be enough in order to extract DNA and multiply in millions of copies. So, next question. The person's fingerprints are necessary in the final step of DNA fingerprinting. Is it true or false? And this is, of course, false. We don't need actual fingerprints of the person when we compare genetic profiles. And here example of the DNA profile of few people. So one person, suspect number 1, number 2, number 3. As you see, genetic evidence collected at the crime scene. This genetic profile is the same as of the suspect number 2. Such profiles are made with help of PCR and gel electrophoresis. We need PCR in order to multiply number of fragments. Evidence can be collected in very small quantities, and in order to for us to be able to see such strips on the gel, we need to multiply those molecules, those fragments, in millions of copies, so we can see such strips with naked eye. Of course, we need some pigments that would stick to DNA, and DNA would be visible for us. Sometimes we need ultraviolet to see these colors. All the people have about 99.999 the same genetic makeup, and scientists can use only 1/1000% in order to differentiate each of us, and actually even much smaller fraction is needed in order to make a genetic profile of each person. Then such profile can be submitted into the CODIS database, and later investigators can easily find if any biological evidence collected at the crime scene similar to any of the profiles in this database. And this is all for today. Thank you for your attention. Please subscribe to my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any, and see you in the next video. Goodbye.